Hi, this is Miss Litton, and we're doing a rewind of Chapter 3 and Chapter 4. Say hi.
Hi.
All right, it's a huge group. Whoa. You will remind me later. Okay, so we're just going to hit the Friday night. It is Friday night. Friday night highlight reel of Chapters 3 and Chapter 4. So the first thing you have to know is organic molecules are built with carbon. Carbon, right? Carbon. Okay. That was terrifying. And if you just put a bunch of carbons and hydrogen together, what do you call it? A hydrocarbon. Good. And a lot of times we burn hydrocarbons as fuel, right? Because there's energy in the bond. And then there are functional groups you can attach onto them to give that hydrocarbon a little flavor. Maybe you make that region of it hydrophilic. Could be an amphipathic molecule, or one region is hydrophilic and one region is hydrophobic. And then we talked about we talked about the 4 important organic molecules. So when you think of the 4 important organic molecules, I want you to think what they are, what their building blocks are, but more importantly, we want to know, where do we see this in cell functionality? Do you see it to store nutrients? Do we see it for immediate energy? Do we see the product? I mean, when I see carbohydrates, lots of things come to my mind. I'm thinking carbohydrates, the formation of carbohydrates is carbon fixation. Makes me think about the process of photosynthesis. Because when we learned this, you didn't know how to build sugars, but now you do, right? So I'm thinking about photosynthesis as a way to make a carbohydrate to reduce CO2 into a sugar. And then conversely, I'm thinking that cellular respiration, very good, Jesse, is a way to release the energy out of the carbohydrate. Do you agree with that? Yeah. Okay. When I see lipids, maybe what comes to mind right away are fats. Okay, or thanks a lot, because you're looking at me. Phospholipids. Phospholipids, and I think about cell membranes. Look, 2 APers walking by, not staying. Oh,
maybe they don't know.
Maybe they don't know. Phospholipids. So I think about cell membranes and how we use cell membranes to maintain homeostasis. We use cell membranes to acquire materials, so we have energy. Yeah. So when I think about lipids, I think maybe phospholipids, or maybe I think about steroids that are in membranes, right? When I see proteins, we haven't learned all of that yet. We haven't learned about protein synthesis, but we will. But for now, we know proteins are really important as what?
Enzymatic.
Enzymatic. It's enzymes. We need enzymes to lower the what? Activation energy, right? And enzymes have an active site that binds with a substrate. We know enzymes are super specific, right, in their function. When I see nucleic acids, I'm going to think, of course, of what? Nucleotides. That's what they're built out of. And I'm going to be thinking about DNA, and DNA, how it codes for RNA, and how that codes for proteins. And we're just starting to learn about that now, right? We're going into the molecular biology, and we're going to go into the molecular biology of the gene. So when you were first learning these, you didn't have that added knowledge yet. So now, that's why I like to do these rewinds, because now you look at it with a different, then it was just proteins, what are they made out of? Now we can think of it as a bigger picture. We can think, ooh, proteins and their structure, when we learned about primary, secondary, tertiary, and quaternary structure, that should be meaningful now because we know enzyme functionality is dependent upon the structure. So if DNA codes for the wrong amino acid, then that protein could have the wrong shape, right? And then it loses its ability to do what it needs to do. So that's why we learned those at the time. So think about that. When we talk about building, we learned about dehydration, synthesis, to join it together. And it doesn't matter whether I'm building a protein, a carbohydrate, or a lipid. I know it happens in that same way, right? I'm going to remove that water to join it together. What's another reaction, another metabolic reaction besides dehydration synthesis that would form water? It's another metabolic reaction that would form water. Yes? Either way. Yes, cellular respiration. So now we can see why a kangaroo rat in the desert never needs to take a drink of water in its life. Because these type of metabolic reactions can generate the water that they need, right, in order to survive. Children, I can't have you talk right in the middle of a review, right? You just go back in the corner if you're going to talk. Okay, so now we can understand that. We know how molecules are broken apart. How are molecules broken apart? Adding water to it, hydrolysis, right? Okay, so we learned that. So now when we look at this reaction, which you remember at the time, right? And we could talk about how, and this enzyme is named for its substrate. So the substrate is sucrose, so now we know sucrase. So we could write an essay if it said, what are some enzymes that you know and what they do, right? I could be thinking about this, digestion. I could be thinking about CDKs. What are CDKs?
Kinases.
Yeah, cyclin-dependent kinases, right? That are activated by what? Cyclins, which will facilitate the cell cycle, right? So I can make those connections now when I think about enzymes. When we look at this sugar, we know the formula, if we simplify it, is what? CH2O. And we understand, remember how we talked about carbo, what? Hydrate, its name, right? We think about the water that's in it. We learned about disaccharides, and here are all of those. We looked at polysaccharides, and we looked at some can be for energy storage and some are structural molecules. So we know the importance of cell walls for plants, that they don't have an extracellular matrix on them like we do our animal cells, because they can be held together by cellulose, their cell wall. And we know that not all cell walls are the same, right? We know that fungi make a different cell wall. What do they make their cell wall out of? Chitin, exactly. So you see what I'm saying? When we're layering it up. Now that you have more information, you want to keep going back and saying, how does that change my perspective of what I learned before? Okay, so we looked at cellulose, we looked at glycogen, how we store energy in our liver. And we store a 3 to 4 hour supply. We looked at chitin as a structural component as well. When we talked about lipids, we talked about glycerol and the fatty acid chains. And we know that those fatty acid chains could be what? Saturated or unsaturated. And then I'm going to go back to cell membranes again, because that's your most recent material. If they're unsaturated, right, and it gets really hot, they could end up kind of coming apart. What kind of helps hold it together? Cholesterol. And conversely, if they freeze, cholesterol would prevent that from occurring, right? Okay. And then we looked at this. Yes, we're going to look at steroids in one more minute, but here is an example. And you know there's glycerol, and this is a great example of an amphipathic molecule, because it's got two different properties. At the phosphate, it's hydrophilic, and at the fatty acid chain, it is hydrophobic. And we know how that sets up cell membranes with the hydrophobic fatty acid chains in the middle and the hydrophilic phosphate groups on the edges. And then stabilized by steroids. Not estrogen or progesterone, but cholesterol. Yeah. All right. And then looking in at proteins, and we look at the different functions. We haven't talked about the immune system so much, but we have totally talked about enzymes now, so we understand that. We haven't talked about, okay, I wonder if it paused it. No, we're still going. Okay, we haven't talked about brain and nerve, but we will. We'll talk about blood. We haven't talked about muscles. We've talked a little bit about cellular messengers, right? And we know proteins. Let's go over the functions of all proteins. They could be what? Channels, carriers, cell recognition, receptors, enzymatic, and cell junctions. Good. And we talked about the different kinds of junctions. Sometimes this can also be this, right? It can be a receptor, or you could have an enzymatic one right next to a receptor. The receptor could trigger the enzymatic protein. Okay, we know about amino acids. Okay.
OFAC Schmuel, please report to the office. OFAC Schmuel, please report to the office for your college meeting.
Okay, Okay, and now, again, more significance, understanding about enzymes and reactions, and it's things that could change it allosterically or denature it. Okay, and then that then brings us to nucleic acids, and nucleic acids are built out of nucleotides. We know the difference between nucleotides, between DNA and RNA, would be what? DNA has the sugar deoxyribose, where RNA has the sugar ribose. DNA has the base thymine, RNA has the base uracil. And when we get into the molecular structure of the gene, I believe that is chapter 12, then we'll understand that a little bit more. Okay, and there's RNA, and then we have, here we can see the importance of hydrogen bonding. We saw hydrogen bonding here between the bases, but where else did we see hydrogen bonding? Water molecules. And we know that most of living cells are made out of water. So understanding the properties of water helps us to understand how our cells function, right? Okay, and then that brought us to ATP and then our summary on that. Now I had questions. Did I ever show you this?
Yeah.
That was my messy one. I had questions in here, and there was also a monster quiz. What one do you want to do? Do you want to do both?
The questions. The questions.
Questions, okay. So I'm going to pause this for a minute while I do the questions.
Yeah,
Okay. Yay!
I did the wrong answer or something.
Yay!
I got something wrong back.
Uh-oh. 4 rings of carbon build a steroid, not a carbohydrate. A carbohydrate is not, you can have a ring of carbon, but if I say you have 4 rings of carbon, then it's definitely a steroid. Okay? Yay. Yay. Okay. Let's talk about this one. When you do dehydration synthesis using 2 amino acids, you form a protein. Is that true? Yeah, dehydration synthesis, you're joining them together. Amino acids, you form water. Yes, dehydration synthesis, you form a water. Take an OH and an H out of it and form a water. When you form fatty acids and glycerol to form a nucleic acid. No, it's fatty acids and glycerol to form a... How could you make that correct? Not an amino acid. Fatty acid and glycerol to form a fat, lipid fat. Do you do dehydration synthesis with polymers to form monomers? No, how would we make it? Monomers to form polymers. Good. Nucleotides to form nucleic acid? Yes, and then this no. Okay. Do you understand that?
Yeah.
Okay. Oh, when you do hydrolysis to a fat, do you produce sugars?
No.
No. Do you produce a fatty acid?
Yeah.
Yes, you get fatty acids. Do you make, oh, he didn't hear me. He's not even in my class, but, you know, I just opened classroom. Nucleic acids?
No.
What?
No.
It says it's wrong even though it's right.
I don't know why it showed up on yours that way. Did you mark all of them?
No, I just marked the one that says it
You marked these 2?
Yeah.
Oh, then it should be, right? Yeah, I don't know why it marked you wrong. And then, but do you make nucleic acids? No. Do you form glycerol? Yes.
Yes.
Do you form water?
No.
No, because you have to use water to break it up, right? Okay, and then not oils, right? Because an oil is a complex lipid. Okay, 8. Which of the following is a DNA nucleotide? You said E. Why is D can't be a DNA nucleotide?
Ribose.
Yeah, ribose takes it out of the running. And if you wanted to fix that, it would have to be deoxyribose. Okay, that is chapter 3. Let's look at chapter 4. Good job, you guys, on your rewind. Okay, 4. I have a few.
This is the cell theory one.
Oh, easy peasy lemon squeezy. We could go right to the quiz questions.
Yeah.
Okay.
Actually, can we try them first before?
What? Well, yeah, absolutely. You want to do all of the quiz questions? Okay, we'll go right here and just see what you know. Good job. I'm going to pause this. Remind me to unpause it. Okay, let's see. I unpaused it. I resumed it.
Oh, I got it wrong.
Okay. All right. So I just want to reiterate, because we skipped over this part. Ladies, you gotta be able to hear. Is that you need large surface area per what? Large for cells are small because you need a large surface area per Volume.
Volume.
Huh, I'm glad I just went over that because you forgot it. Okay. All right. So then we went into the structure of prokaryotic cells. Remember all of that?
Yeah.
Okay.
Corn dog.
Corn dog, hairy corn dog. We compared the 3 domains. Okay. Then here, let's see how you do on this.
It's also one question.
I'm going to give you 10 seconds. So you better answer it. 10.
Answered.
I hear your voice saying
Oh, what structure is characteristic of prokaryotic cells? Nucleoid. Remember, prokaryotic nucleus. And nucleolus is inside the nucleus of a eukaryotic cell, and mighty mitochondria only in eukaryotes. But keep in mind, they're still doing cellular respiration. It's just not contained within an organelle. Okay, that's why. All right, then we talked about the endosymbiotic hypothesis. Remember that? Did I pause? No. Okay, we talked, are you comfortable with that?
Yeah.
Okay. And then we went on and talked about plant cells, animal cells. We went through all their structures.
Golgi.
So many. You know it all.
That's so weird.
Because
We love you, Miss Lynn.
I love you right back. Okay, here you go. 13 questions.
I
Oh!
remember this. I remember doing this. Which organelle is the...
Okay, here we go. Let's see how we did. Yay! 1. Wait, I don't want to go too fast. Do you know, are you good with that?
Yeah.
Yeah.
So I'm right in your way. I don't know how to do this. I don't know if I should move this. Sorry. I don't know why it goes up. I don't know. Okay, anybody trouble with this one? No?
No.
Okay. Uh-oh. Which organelle processes, packages, and distributes its Golgi apparatus? One person said ER.
I put that back then.
Okay,
Yeah,
all right.
no.
So you good now?
Yeah.
You fly. Okay. We good with this one?
Yeah.
Okay. Okay. Which organelle supports, protects, is permeable to gases and water so they can enter and exit the cell? Yes.
Yes.
Cell membrane doesn't do that. It doesn't, it's not about support, it's not really about protecting. Your cell wall is about protecting and supporting. And those were the lead words that came out, so that's why you would pick that. Permeable to gases, plants have to have O2 and CO2, right? Yes? Okay, and water so they can enter and exit. Okay, good. Yay! Anybody think of something else? Good? Okay. Good?
Yeah.
Good? Oh no, we not good. Okay. So which organelle is a multi-purpose organelle? Stores, transports, support, turgor pressure, should have given it away. Turgor pressure in plants. That's the vacuole that does that. Cell wall does not store. Okay, cell wall does not store.
Doesn't the cell wall create turbo pressure?
The cell wall pushing back, yes, but only if the vacuole is pushing on it, right? So, but what you want to do is you have to go, what applies to everything? And a cell wall doesn't, you don't think of that as storage. Okay? Okay, and 10, perfect. Was anybody thinking of something else? Okay, 11, questions? What, uh-oh. Which of the following has 9 sets of 3? Centrioles, things that look like churros, which we're learning about now that are at the centrosomes of microtubule organizing centers, MTOCs. 9 groups of 3, and so too do the basal bodies. The basal bodies anchor cilia and flagella. The basal bodies anchor the cilia and flagella. So maybe one came from the other. And then it's the cilia and flagella that have 9 sets of 2 with 2 singles in the center. So that's not a comprehension thing. That's more of a, oh, okay, that's what it is. But what you want to see is that the cilia and the flagella that has the 9 sets of 2 and 2 singles becomes the basal body where it gets anchored and it switches to 9 groups of 3. and that also animal cells don't have any, I mean, animal cells and some protists have the centrioles, but plants do not have any centrioles. Yes,
Could you go to 10 for a second?
I can.
So, I know why it's ribosomes.
Right.
But if it had said rough ER, where the ribosomes are located, how would we
I wouldn't have
differentiate?
picked ER or rough ER if ribosomes was there.
Okay.
Because not all ribosomes are on the ER. Free floating ribosomes are what make the proteins for peroxisomes. So not all ribosomes are on the ER, but you have to have a ribosome to do protein synthesis. Yeah. If you had nothing else, if you had nothing else to choose, then you could choose that. Okay? Any other questions on that one? Okay, you did it all. I'm so proud of you. That was it. That was the last set of questions.
Go us.
It helps
Go
to
you.
have a small review group.
Yeah, it does, but I'm really proud of you. Stranger dangers. Hi, sweetheart. I'm really proud of you for coming in even when you didn't have a quiz or a test. That shows extreme discipline, and you took care of reviewing 2 chapters for your final. Yes? Yay! And everybody listening to this review is thankful, because if you weren't here, it wouldn't have happened. Okay? Good job. If you're tired, have a piece of toast. 21 minutes of fun.